Neurotransmitter tests do not measure the amount of neurotransmitter levels in your brain and are a total waste of money. Firstly, I'm going to tell you why neurotransmitter tests don't measure the amount of neurotransmitters. Then I'm going to explain why even if they did, it still wouldn't help us to solve our mental health problems. And finally, I'll tell you what to do instead. If you haven't already had a neurotransmitter test, watching this video could save you several $100s. If you've already spent your money, you'll just have to write it off. So why don't neurotransmitter tests work? Well, firstly, the neurotransmitters in the brain don't actually leave the brain and end up in the urine. They are contained within the blood-brain barrier that encircles the brain. What so-called neurotransmitter tests actually measure is the waste products of neurotransmitters after they have been broken down. In biological speak, you would say the metabolites, or the breakdown metabolites of the neurotransmitters. Let me draw you a diagram. So in this classic diagram of a synapse, the top neuron is making serotonin shown in blue and dopamine shown in red. The neurotransmitter is released into the synaptic cleft. That's the space between the two neurons. The neurotransmitter travels across the synaptic cleft and attaches to receptor sites. Once attached, it stimulates the release of G proteins, which then stimulate second messenger signals within the receiving neuron. At the same time, the excess neurotransmitters are being broken down or metabolized. In the case of serotonin, it's broken down into something called 5-HIIA, and that's what's measured in the urine tests. In the case of dopamine, it is broken down or metabolized into something called HVA, and that's what the urine tests measure. At the same time, the unused or unbroken down neurotransmitter is reabsorbed back for recycling purposes into the transmitting neuron. The transporter protein that transports dopamine back into the sending neuron is called DAT or DAT. In the case of serotonin, the transporter protein that recycles serotonin is called SERT, S-E-R-T. Now, back in the 90s, we believed that the way that antidepressant drugs like the SSRIs worked was because they inhibited these transporter proteins, causing the neurotransmitters to linger in the synapses longer and therefore stimulate more second messenger signaling in the receiving neuron. Now let's say that your test shows high levels of HVA in your urine. Remember, HVA is the metabolite that dopamine is broken down into. Do the high levels of HVA mean that our neurons are making the right amount of dopamine, releasing the right amount of dopamine, but it's being broken down too quickly? So we end up with low levels of dopamine in the synapses and possibly depression or ADHD. Well, no, it doesn't necessarily mean that we have low dopamine in our synapses. Seeing high levels of HVA in the urine could mean that we're making way too much dopamine and our brain is attempting to break down the excess, resulting in high levels of HVA in the urine but still high levels of dopamine in the synapses. And the third explanation as to why we see too much HVA in the urine could be that our brain is making slightly too much dopamine, but it is breaking down the excess perfectly adequately so that the level of dopamine in the synapses is perfect. So you see, there's no way of knowing if the high level of HVA in the urine means that your dopamine levels are too high, too low, or just right. And you would have the exact same problem if the level of HVA in the urine was found to be too low. It doesn't tell us if the levels in the synapses are too high, too low, or just right. Unfortunately, we just don't have any way of accurately measuring the amount of neurotransmitters in the synapses. It's estimated that there are from 100 to 500 trillion synapses inside the brain. So these are absolutely tiny, microscopically small structures. And there's so many of them. What would you do? Sample a few and then take an average? And that's not the only problem with measuring neurotransmitter metabolites in the urine. You see, there's more neurotransmitters made in our gut than there are made in our brain. And the waste metabolites of the gut neurotransmitters are also going to turn up in the urine. So what are you actually measuring here? Then there are the transporter proteins that remove the neurotransmitters from the synapses, but do not metabolize or break them down. this effect doesn't even show up in the urine. And we now know that mental health conditions involve totally separate problems from the level of neurotransmitters inside the synapses. The actual number of synapses and the number and type of receptor sites inside the synapses, all of these things play a role in causing mental health problems. We need to stop thinking about mental illness as just a chemical imbalance in the levels of neurotransmitters in the brain. That understanding had its heyday back in the 1990s. By the early 2000s, anyone who still believed in that explanation of mental illness was falling behind in their understanding. Talking a bit more about the reabsorption and the transporter proteins, we now know that some people have excessive amounts of the transporter proteins, so they may release enough neurotransmitter into the synaptic cleft, but end up with low levels of the neurotransmitters crossing the cleft and attaching to the receptor sites, because they have too much of the transporters removing the neurotransmitters too quickly from the synaptic cleft. Genetics have now identified some of these variations. So some people have the genetic variation that they overproduce the DAT, dopamine transporter protein, and this could result in depression or ADHD. Some people have the genetic variation so they overmake the serotonin transporter protein called SERT, S-E-R-T. This could create a type of low serotonin depression. It'd be involved in poor appetite control, particularly craving carbohydrates, particularly waking early. Back in the 1990s, it used to be believed that the SSRI antidepressant drugs, selective serotonin reuptake inhibitors, worked by inhibiting the SERT protein transporter, and this produced the antidepressant effect. And the same mechanism was thought to work with the SNRIs and other types of antidepressants. But by about 2000, it became obvious that this explanation of how these drugs work just had to be wrong. The reason this was obviously wrong is that these drugs would actually start inhibiting the transporter proteins as soon as they reached the brain, which would be within about an hour or so of taking them. But we all know antidepressants often take 3 to 6 weeks to produce any effects. So there just had to be another biological explanation of how these drugs worked. It couldn't be that they were inhibiting the transporter enzymes. Sorry, I meant transporter proteins, not enzymes. Later I'll give you the updated explanation of how we now believe the antidepressants work, which explains why they take 3 to 6 weeks to start working. So let me just summarize what I've said so far. Testing the level of neurotransmitter metabolites in your urine does not accurately indicate levels of neurotransmitters in your brain or synapses. Firstly, it actually picks up the neurotransmitter metabolites that are coming from your gut. And secondly, it has no way of informing you if the levels of neurotransmitters in your synapses are too high or too low, because your neurotransmitter transporter proteins may be removing the neurotransmitters from the synaptic space too quickly or too slowly. That's also possible. And the neurotransmitters that are reabsorbed by the transporter proteins don't show up in the urine at all. But there are even more reasons why neurotransmitter tests are a total waste of money, which I'll go on to explain. So we now know that what's just as important as the amount of neurotransmitters inside the synapses is the actual number of synapses. We know that chronic inflammation in the brain prunes away synaptic connections, and if the rate at which inflammation is pruning away synapses is faster than the rate at which your brain's neuroplasticity can replace and grow back the lost synaptic connections, then you're going to end up with a net loss and a reduced number of synapses. And it turns out that the pruning effect from inflammation is particularly aggressive in the key structures of the brain that control mood. So you can have depression not because you have too little serotonin or dopamine inside your synapses, but because you've got too few serotonin and dopamine synapses left to hold and transport your neurotransmitters. Let me show you a diagram to illustrate that. In this illustration, we can see the devastating effects of inflammation. This first slide is a normal healthy neuron. It's grown a good number of branches, and if we blow up the side of a branch, we see these little bumps. They're called dendritic spines. Each one of these is a potential synapse and capable of growing a new synapse. In the middle, this is the effect of exposing the brain to chronic inflammation. The inflammation has pruned away the branches, what are called dendrites, and pruned away the dendritic spines, resulting in a net loss of synaptic connections. This would produce a depressed state. In this experiment, after the source of inflammation, which was actually injections of cytokines, was removed, and high doses of lithium carbonate were administered, which boosts neuroplasticity, particularly boosting this chemical here called BDNF, you see that the neuron, which is not dead, is perfectly capable of growing back a healthy amount of branches and a healthy amount of dendritic spines. If you have the situation in your brain that you've just seen in that diagram, a much better and more advanced treatment solution would be to reduce inflammation and boost neuroplasticity to help grow back lost synapses, as opposed to just trying to boost the amount of serotonin or dopamine inside the diminished number of remaining synapses. It was about 15 years ago when I first saw that diagram and finally understood this phenomenon of inflammation pruning synapses faster than we can grow them back. I started putting together holistic treatment approaches to reduce inflammation and boost neuroplasticity that included supplements, diet, brain training exercises, sleeping techniques, the whole package. And the therapeutic results were just phenomenal. It significantly changed my ability to control my own bipolar disorder. And I've used these techniques to help many other people with bipolar, depression, ADHD, recovery from traumatic brain injury, and other conditions where increased inflammation is attacking brain structures. Now, do you remember I said I'll explain why we now believe antidepressant drugs take 3 to 6 weeks to work? Well, this is one of those reasons or explanations. You see, it turns out that the modern reuptake inhibitor antidepressants also have anti-inflammatory and neuroplasticity boosting effects. And this explains the time delay. Because as you start reducing inflammation and boosting neuroplasticity, it does take several weeks of doing that to grow back enough synapses to have an antidepressant effect. There's actually another exotic, unusual explanation of why modern antidepressants take several weeks to work, which I'll give you in a minute. But let me give you another super important new understanding that goes beyond neurotransmitter balance, which is the importance of the receptor sites. Receptor sites are what the neurotransmitters attach to on the receiving neuron. And there are several types of dopamine and serotonin receptor sites, each with different functions, and they are concentrated in different parts of the brain. Some are stimulating or have stimulating effects, others have inhibiting effects. In bipolar disorder and schizophrenia, for example, it's believed that there is an overexpression of a type of dopamine receptor called the D2 receptor, and it's believed that many of the drugs with anti-manic and antipsychotic effects work by changing activity in the D2 receptors. What this means is the amount of dopamine inside the synapses might not really matter that much. What might matter much more is the type and the number of receptor sites on the receiving neuron. And there's another important thing. The neurotransmitters just attach to the receptor site. They don't actually pass through into the new receiving neuron. When they attach to the receptor site, they trigger or set off signals that run along inside the receiving neuron. These are called the second messenger signaling system. And we now know that there can be imbalances in the second messenger signaling systems. When a neurotransmitter attaches to a receptor site, it releases these proteins called G proteins. They then stimulate second messengers. And it turns out that we can have imbalances in our G proteins. There are GS stimulating proteins that stimulate second messengers, and there are GI or G inhibiting proteins that inhibit second messengers. If you have an imbalance in the stimulating versus inhibiting G proteins, you're going to either get too much signaling inside your neurons or not enough. Too much would result in things like mania, psychosis, that kind of inability to fall asleep, switch the brain off and fall asleep that we get during mania and psychosis. And too little second messenger signaling can result in depression. This overfiring or underfiring of the second messengers is not to do with the amount of neurotransmitters in the synapses. Understanding that the activity in the brain can be sped up or slowed down or increased or decreased because of imbalances in the second messengers is another reason why we need to move beyond thinking about neurotransmitter levels. I said earlier that there was another new, exotic or unusual theory that explains how antidepressants work. It's been discovered that during depression, the G proteins end up detached from our receptor sites and end up sitting on rafts or blobs of fat elsewhere in the cell membrane. The way I visualize it is that the G proteins have left their workstation and gone on vacation. They're now floating on a lipid raft. And what was discovered is that the modern antidepressants, the SSRIs and the SNRIs, gradually over several weeks accumulate on these lipid rafts and displace the G proteins off the raft back to their workstation attached to the inside of the receptor sites. And it takes several weeks for the antidepressants to concentrate on the lipid rafts. So again, this is another explanation as to why do antidepressants take 3 to 6 weeks to work. Can I ask you to do me a favour? If you're finding this information interesting or helpful, please give me a thumbs up. This not only helps me to understand what type of content you would like me to make more of, but it also helps to promote this channel and this information to a wider audience of people with mental health problems who may benefit from it. So where does all of this leave us? If neurotransmitter tests don't tell us which neurotransmitters may be too low in depression or too high in mania and schizophrenia, how can we work out how to treat our neurotransmitters? The solution I use to treat my own bipolar depression and hypomanias, and then in my practice, was to learn to read the signs that indicate which neurotransmitter has gone out of balance in the brain. So for example, dopamine runs the circuits in the brain involved in the pursuit of reward and pleasure, in motivation, in excitement about doing things, especially new things. So if you observe in yourself an increase in that type of behavior, it indicates an increased level of dopamine activity in your brain. Alternatively, if you observe yourself having diminished or no motivation or enthusiasm or interest in things that usually used to interest you and give you pleasure, if you have diminished drive and excitement, this looks like dopamine activity in your brain has become underactive. There's a name for this state, it's called anhedonia. I also call it a low dopamine depression. Now, for all of the reasons I've just explained, it doesn't tell you whether or not you're not making enough dopamine, or you're making too much of the dopamine transporter, or does it mean that you have too few dopamine synapses left, or too few dopamine receptor sites, or are your G proteins either out of balance, not enough of the stimulating and too much of the inhibiting G proteins? Or have your G proteins left their workstation, gone on vacation, and are floating on lipid rafts? I know it's super frustrating not to have precise ways of knowing which of these possibilities is causing the anhedonia or other mental health problems. But that's the situation. At this point in time, we just don't have any testing systems that you can buy to work out how your neurotransmitters, synapses, or second messengers are working. But just because we can't test it in advance doesn't mean to say we can't gradually figure out solutions. What I do is just start applying different treatment strategies and see which one works. Something you can always start with is immediately eliminating all unnecessary sources of inflammation. You can start feeding the brain what it needs to grow back synapses, especially EPA and DHA. Then when you've made some progress with that, you could try boosting the production of dopamine with the amino acids tyrosine and other herbs that boost dopamine. And you can combine the things that boost the production of dopamine with things that inhibit its breakdown. At the same time, start doing things that boost neuroplasticity to grow back brand new synapses. And take things that specifically protect the dopamine synapses and neurons from damage, such as skullcap, lithium orotate, bacopa. Now you might be thinking, well, how do I know which treatment strategy is actually working? Well, yeah, you're right. You actually don't really know precisely, but there's two answers I've got to that problem. One is sometimes you do get an indication. You might introduce a particular new strategy and notice, oh wow, that's really making a difference. And the other thing is, what's more important than knowing which of the treatment strategies worked is actually just getting a result. If you were depressed and you tried 4 or 5 antidepressant treatment strategies all at the same time and you ended up not depressed, do you really care which one of the treatments, or was it the combination that worked? If you're a research scientist, then you absolutely need to know which one of the techniques worked. Researchers would actually hold back potentially helpful treatments while they test one thing at a time. That's not my job. My job is to get as many of my patients well as quickly as possible. So my job as a practitioner, or I could say our job as patients, is to throw everything we possibly can at the problem and hope that we get a result. Something really important I should just add here is if you have a bipolar or schizophrenic brain, you need to be super cautious boosting dopamine. In fact, most psychiatrists would say don't boost dopamine because it could flip us up into mania or psychosis. The problem I have with that is there are situations where if you don't boost dopamine, you end up living with some level of anhedonia. So the solution I use with my own bipolar disorder and with many of my patients is the first thing you do before you boost dopamine is you must put in place really strong and robust protections against dopamine going too high and inducing mania or psychosis. That may require pharmaceuticals, supplements, and special sleeping techniques. Then, cautiously, once you set a protection against dopamine going too high, causing mania and psychosis, you could carefully experiment with just lifting dopamine enough to overcome anhedonia. Another thing, which is a safer strategy, is rather than trying to boost dopamine directly, boost dopamine neuroplasticity to try to grow back a healthy and well-balanced dopaminergic system. Now, moving on to serotonin. Serotonin soothes pain and distress. It gives us a feeling of calm and happiness. It enables the psychology of tolerance and empathy. One of the ways that we know this is because of the effects of the street drug ecstasy. Ecstasy floods the brain with serotonin and not only induces a feeling of happiness, but also changes people's behavior to become more tolerant and empathic. Now, if that's what serotonin gives us, when you take serotonin away or lower levels of serotonin, you end up with not a numb, depressed feeling like you get with dopamine, but a very intensely painful type of depression. Lots of crying, there could be irritability, short-temperedness, aggressivity. When you know what a neurotransmitter does in the brain, it's quite easy to learn to read the signs that indicate if you've got too much or too little of it. Other things that serotonin is involved in is getting enough sleep. So if you are having difficulty sleeping, especially waking early in the morning, feeling anxious, this is a strong indication of low levels of serotonin activity in your brain. Very different from low dopamine depression, where we end up sleeping too much. In one of my next videos, I'm going to go into more details about how to read the signs and symptoms that indicate which neurotransmitter has gone out of balance. Actually, you could let me know in the comments if you'd like to see a video on either how to read the signs that tell you which neurotransmitter has gone out of balance, or a video going into details of the role of dopamine in the bipolar and depressed brain. And when it comes to treating low serotonin, well, it's pretty much the same situation as with low dopamine. We don't know exactly what the cause is and there's no way of testing, but there is one treatment target that really stands out with restoring healthy serotonin levels, and that is reducing inflammation. This is because serotonin metabolism or production is super sensitive to inflammation levels in the brain. Just a small increase in the level of inflammatory signaling molecules called cytokines increases the production of an enzyme called IDO. Serotonin is made from the amino acid tryptophan, and the enzyme IDO shifts the production of serotonin down another pathway and instead turns tryptophan into a toxic chemical called quinolinic acid. If you want to research this further, search Google for TRICAT. There's the spelling. TRICAT stands for tryptophan catabolites. Put a plus sign and then depression or mental health problems. Another thing you could search for is kynurenine. You see the spelling there. Kynurenine plus depression or kynurenine plus bipolar disorder, and you'll see plenty of information. Kynurenine is an intermediary between tryptophan and quinolinic acid. I should have said earlier, you can also find lots of scientific papers discussing the G proteins, second messengers, and other science topics I've mentioned if you want to do further research. Understanding just how sensitive serotonin metabolism is to increased levels of inflammation has turned out to be incredibly helpful. It tells us that to fix serotonin activity in the brain, we must first eliminate all sources of inflammation in the body. I go into a lot of detail about where inflammation comes from and what we have to do to eliminate it in my video called Bipolar Mania Causes and Solutions. Even if you don't have bipolar, the section on inflammation in that video will still apply to you. We now know there are about 12 common causes of inflammation that we should look for and fix if we have a mental health problem. There's infections, exaggerated stress responses, inadequate sleep, a leaky gut that allows gut bacteria to enter into the bloodstream and activate the inflammatory immune response, an unhealthy diet, allergies, and other things. If you don't eliminate inflammation and you still have high levels of the IDO enzyme, and then you go and take tryptophan to try to increase serotonin production, you may actually end up just increasing the production of quinolinic acid and actually end up aggravating the problem. I've seen this many times in my practice, and it's a telltale sign that we need to hold on trying to boost serotonin until we've first eliminated all sources of inflammation. You could temporarily try to get around the problem by taking 5-HTP instead of tryptophan, because 5-HTP is not converted into quinolinic acid. But this is really only a temporary solution. Moving on, there's one last thing that I want to mention that totally kills off the simplistic model of mental illness being caused by a chemical imbalance in the neurotransmitters. And that is that in the brains of people with depression, bipolar disorder, schizophrenia, OCD, and ADHD, there are actual changes in the structures of the brain, in the wiring of the brain. For example, in the brains of people with mental health problems, there's often overactivity in a structure called the amygdala, and this causes exaggerated stress and emotional responses. There can be poor functioning in a structure called the ACC. The ACC is very much involved in depression, emotional processing, and many of the features of ADHD, like poor time management, time blindness, poor concentration, and other aspects of executive function. And we cannot significantly improve the function in these circuits and structures by manipulating neurotransmitters. The treatment to use to work on this issue is meditation brain training exercises. So this is something totally different to treating mental health problems by rebalancing neurotransmitters. If you'd like to learn more about the structures and circuits involved in mental health problems, watch my video called Solving Anxiety. Even if you don't have anxiety, there's a lot of information in there about depression and bipolar disorder. I'll put the links to the videos I've mentioned in the description, or you can find them on my channel page. In my practice, I've developed specific meditation brain training exercises to target the structures and circuits involved in mental health conditions. I have treatments for recalibrating our stress responses, for healing the gut and other sources of inflammation, boosting neuroplasticity, but also regulating neurotransmitters, boosting them in depression, ADHD and anxiety, and attenuating or reducing them in bipolar, psychosis and schizophrenia. Rebalancing neurotransmitters is still important and a great way to help treat a mental health condition. But just don't waste your money on testing the level of neurotransmitter metabolites in your urine. If these subjects interest you, you can find a lot more free information on my other YouTube videos and on my website, balancingbrainchemistry.co.uk. And if you want to take it to the next level and get my professional help to put together an individual holistic treatment program to work on the root causes of your mental health problems, I do one-to-one consultations online. You can book a consultation on my Calendly link. Just put calendly.com/PeterSmithUK into your web browser, pick a time, go through the payment process, and I'll look forward to seeing some of you there. I hope you find these videos helpful. I wish you every success with your recovery. Bye for now.